So today we're going to talk about the aging of the immune system, and that will include a brief introduction into what the immune system is and what it does, how it ages and why that might be a problem for longevity, and also about the research that we do specifically in my laboratory about trying to understand the aging of the immune system and trying to find the ways to correct and improve the functioning of the immune system in the older age. So the reason why we have the immune system is to defend us against infectious microorganisms that we also call microbes. And those microbes or microbial pathogens are varied and numerous. There's a lot of them. There are bacteria that live on the skin, cause skin infection. There are other bacteria that can live inside of us that can cause nasty things such as diarrhea or autoimmune diseases by virtue of trying to look like your parts of your heart or parts of your joints. There are lots of different viruses, such as many of the common ones, including influenza and so forth. There are fungi, which very often cause infections as well. And then there are parasites like the different protozoans, which are really single-cell microorganisms that can get into your cell and enslave it from the inside. And that's the case, for instance, in the case of malaria or African sleeping sickness. And there are larger parasites also, like the worms, that can really reach very large size and that would live typically in your intestine or in other parts of the body. And the immune system is charged in fending off all of these infections. And in order to do so, the immune system is positioned to intercept microbes wherever they may try to enter the body. It's really strategically positioned all over the place, all over the body. And it is organized in lymphoid tissues so that it can efficiently survey the organism, and the primary areas of surveillance are going to be the skin and the mucosa, the mucosal membranes, because those are the parts of our body that are in touch with the outside environment. In order to deal with infection, we have developed two types of immunity. There is a very evolutionarily conserved and very effective innate immunity. That is the one that we are born with. We don't have to do anything to really activate it very much. It is immediate. It's like a blunt hammer. It slows the invader down. It also has another task, and that is to alert and initiate the reaction of adaptive immunity. And the adaptive immune system is the second immune system that only really exists in vertebrates. It doesn't exist in non-vertebrates. It has been evolved to specifically defend multicellular organisms that are very complex. And it's very highly microbe specific. It is laser-like in its action, and it orchestrates elimination of the microbe in a very, very precise manner. So the basic concepts that I would like to introduce to you are that the immune system has really evolved to discriminate between something that is not infectious and that is self, that is our own body, from something else that is infectious and is not self, and by definition is therefore fulfilling the criteria for a microbial pathogen. And the idea is that the immune system, once it recognizes something as both infectious and non-self, it will attack it. By contrast, it will not attack non-infectious self in any way, shape, or form. It's really trained to try to avoid autoimmunity. Microbial pathogens are first held back by physical barriers, such as skin and the mucosal surfaces, and also chemical barriers in the form of different chemicals that flow in the bodily fluids that bathe the mucosa, for instance, like the enzymes such as lysozyme or the low pH in the GI tract. Once these barriers are breached, then the invaders are first detected by the innate immune system. And as they're detected by the innate immune system, they will contain, the innate immunity will contain the first burst of the pathogen, and it'll slow them down. It'll also, as I said, alert innate adaptive immunity to go and detect the presence of infection and eventually eliminate it. Adaptive immunity, more specifically T and B lymphocytes, will come in then and clear the infection. And this next slide really illustrates the components of the immune system. We have all of the anatomical and physical barriers in the form of the intact skin. Many of our mucosal surfaces, such as those in the lungs, are equipped with a ciliary apparatus that constantly flows and essentially expels the junk out of our respiratory system and many of the other systems. There's low pH in the stomach and so forth. And then we have this innate immunity, which is composed of cells and free-floating molecules. Cells consist of macrophages, eosinophils, neutrophils, and dendritic cells, and so forth. And this is the cellular component of the innate immunity. And then we have molecules that are present in bodily fluids, certainly in the blood and other bodily fluids, such as the complement system and the antimicrobial peptides and different LPS binding proteins, the C-reactive protein. And then on the side of the adaptive immunity, we have cellular immunity, which is composed of T and B cells. And then B cells do one other very important thing, and that is that they make antibodies. And once they make them, antibodies will be also secreted and will become part of the humoral adaptive immunity. Antibodies will be present at a high concentration in our blood, and they will be able to then bind the invaders and neutralize them, and also make them susceptible to being sedimented away and eaten away and degraded and so forth. So the important thing is that we also need a balanced immune system. By exposure to antigen, we have the division between the B and T cells, where the naive cells exist in our bodies that have never seen antigen. They're very diverse, they're ready for any potential challenge, they defend against new infection, and they are the ones that are in charge of eliminating and dealing with pathogens that we have never seen before. They're sort of like this large army that we have in our bodies that's ready for any possible invasion. They typically live in organized lymphoid tissues, most notably in the lymph nodes, and they wait for their specific microbe or their specific bug to come, and should it come, they are poised to react in a very organized manner. Memory cells, on the other hand, are the antigen experience cells. They're the ones that have grown out of the naive cells after the naive cells have seen the bug. And through that process of antigen recognition and division and differentiation, memory cells are now hardwired to very promptly respond to challenge. They don't need very much time to respond. Naive cells, on the other hand, need to multiply, need to proliferate. There's relatively few of them that are specific for any given bug because we can't be only filled with lymphocytes. And so in order to accommodate all of the lymphocytes that need to defend us against any possible invasion, we have to have relatively few of them. But they are endowed with this property of massive proliferation, and once they proliferate and differentiate into competent defenders of the organism, some of them will survive for many decades as memory cells. They provide immunity against pathogens that have attacked us in the past and that therefore are likely to be around in the environment to attack us again. And they're so effective that very often we don't even feel that there is this second infection and second attack. They will eliminate the bug before it can cause any disease or sickness. They're actually the basis of vaccination. What we do when we vaccinate, we turn some naive cells into memory cells in a deliberate fashion, and then we have the cells that are ready to defend us immediately. The important point that I'm trying to make here is that we need to have some naive cells, we need to have some memory cells. This is the balance of the immune defense. Our immune system works best when those two components are balanced. And that is one of the problems that occurs in aging, that there is no balance anymore between these components. We're going to talk about that. So let's talk a little bit about what happens when the microbial infection actually occurs. How is the innate system communicating with the adaptive immune system? That's very important because that's one of the key things that breaks down in aging, and we'll address later. So everywhere around our body, underneath our skin and underneath all of the mucosas that we have, there are sentinel cells called the dendritic cells. And these cells are particularly suited to eat up, gobble up, and chop up and degrade any foreign invader that can show up. So here we have this immature dendritic cell that's sitting in the tissue, and in these little diamonds we're representing the virus. So this is the viral infection. But almost momentarily after the virus ends up in the tissue, the dendritic cells will eat it up. They will gobble up the virus, and they will start chopping it into the pieces that the lymphocytes can see. And at the same time, this will be the signal for the dendritic cells to mature. As they mature, they will get a license to leave the skin or the mucosa and to go to the nearest lymph node or the nearest organized lymphoid tissue. And the reason why they go there is that this is where the naive T cells live, and the naive B cells are there as well. But particularly, it's critically important that the dendritic cells talk to the naive T cells, and as they talk to the naive T cells, they will alert them to the presence of this virus, and they will find those that are specific for the virus. And at that point, there will be antigen-specific lymphoid activation. One T cell is going to multiply so that it will create 1 million of T cells that are identical to it. And it will also differentiate so that it will become this very angry, very well-armed cell that is now ready to go back. And what it does, it actually really literally traffics out of this lymph node once it has been activated. And the B cells do the same. The B cells get their signals a little differently, but the principles are the same. They will start making the antibodies, and both the antibodies and the B cells will leave the lymph node. The important thing is that the T cells will now migrate back to the tissue where the virus originally lived and where it's trying to multiply, and they will go in there and deal with the virus. They will produce a lot of antiviral drugs, antiviral components, antiviral chemicals. They will also be able to kill those cells that have been infected with the virus, because what the virus does, it really gets inside the cell and uses the cell as the factory for its own multiplication. And the T cells will go in there and they will often say, well, this cell is too sick to be saved. We're just going to kill it so that it doesn't produce any more virus. And so this actually closes the circle, and at that point the virus is controlled in the tissue and it's completely eliminated, and we no longer have the disease state, and we're cured of the infection. Some of these T cells, as I mentioned, will survive and become memory cells, so that should the same virus attack again, we'll be able to defend very quickly against it. So why are we studying immunity in aging, and how does immunity or immunology help us understand what's happening in aging? Well, there are some very practical problems that arise with the old age when it comes to infections. You know that we had major problems with infections back throughout the centuries, all the way up until the beginning of the 20th century, in the middle of the 20th century, where at least we were able to control infections in kids and in adults. But unfortunately, we are still not very good at controlling infections in older adults, because in older adults, there is high susceptibility, particularly to emerging and new infections. And once older adults get the infection, they're really very likely to get quite sick, and very often also to die from those infections. And to make things worse, they also have poor response to vaccination. So why is this happening? So here are some numbers that can illustrate to you how important this is. So in the US alone, about 30 to 40,000 people die every year of flu. And that's only one infection that affects older adults. And over 95% of the people that die are over 65 years of age. So it's telling you that we really have a problem at the level of older adults. You know that population of people over 65 is very rapidly growing, and so this is becoming an increasingly acute problem for industrial but also developing societies. And I told you already that there's this double whammy of immune aging. Vaccination is similarly inefficient in older adults, and only roughly about one in three people will really benefit. Only 1 in 3 people over 65 will really benefit from yearly flu shots. The problem is that we don't know which ones will benefit from the flu shots, and therefore everybody should be taking the flu shots. Now, what I'm going to be talking to you in the rest of the talk is what goes wrong and how can we make sure that close to every person benefits from the flu shots. And that will require engineering new flu shots, as well as doing some of the rejuvenation therapies and techniques that is also the topic of the work of the SENS Foundation, who has supported parts of this work. So there's a description of age-related changes in innate immune function, and these are still incomplete descriptions. We still don't understand very well what changes in the immune system. Certainly we know that there are some clear defects in the granulocytes and so-called NK cells, which are some of the cells that are in charge of also killing other cells that might be too sick or gobbling up and destroying bacteria in particular, but also viruses in some cases. There's also a change in subset distribution in macrophage function. Macrophages are another cell type that's very good at eliminating invaders. And finally, there are changes in dendritic cell structure and function that are still incompletely understood and controversial, and we need more research. We'll talk a little more about that down the line. What we do know, however, is that adaptive immunity is very much affected with aging. T and B cells are certainly affected. There's a decline in adaptive immunity, and B cells are affected. T cells are even more affected. They're very consistently affected in people over 65. And common age-related changes in both B and T cell lineage include decline in the production of new naive lymphocytes from bone marrow or the thymus. So bone marrow makes the B cells and the thymus makes T cells. And so there's a general shift in making other bloodlines from the early precursors, but not lymphocytes. So lymphocytes are, for some reason, not made all that well as we grow old. And as a consequence of that, there's a replacement of naive cells with memory cells. There's certainly more memory cells in terms of the fractions. There's also an accumulation of certain cells that look like they're aged. They're very difficult to kill. They kind of hang around. They do very different things than what they did in the young age. Both T and B cells have to be very diverse in order to be able to respond to any possible infection. And one of the things that goes wrong with aging is that the T cell and B cell diversity also goes down. So not only are they reduced in numbers in terms of the naive cells, but we don't seem to have quite the right amount of the right cells. And we'll talk again about this very, very specifically because that's part of the research that my laboratory is very interested in. And there's also a reduced ability of naive lymphocytes to respond to antigens. So once they're stimulated, they just don't jump as high and as well as they did in youth. So for the B cells, all of these changes result in making antibodies that are of worse quality. They don't bind their bugs as well, they don't interact with cells appropriately, but also they're reduced in quantity. So they're both qualitative and quantitative problems with B cells. What doesn't help B cells here is that normally B cells, as they develop into the optimal antibody making cells, they need T cell help. So in order for the B cells to make good antibody, they need to get the signals from T cells, and T cells themselves are very much affected and will contribute to the decline of this B cell function. So in the T cell arm of the immune response, we know that several defects exist. T cells themselves at the cell level have intrinsic defects, so they cannot really receive and react to the signals as well as they did in youth. T cell partners that are supposed to tell them that there is infection. I told you that dendritic cells pick up antigen and talk to the T cells and activate them. These partners in regulating the response against microbes also age, and therefore they are likely to produce initial signals that might be delayed or not as strong. And that's another problem in T cell activation. But also the other problem that I've mentioned a little bit before is that there is a problem in T cell population balance. I told you that there is declined production from the thymus, which makes new T cells. There's also impaired maintenance. There's lifelong usage. There is repeated and persistent stimulation, particularly by those infections that infect us in youth and then live with us until we die. Herpesviruses are a good example of the bugs that are not eliminated after infection, but rather go dormant and then reactivate. There's an accumulation of certain clones at the expense of the other clones, so there's a disbalance that will affect the diversity. And also there's not enough of some cells the quantitative problem and not enough of the right cells, the diversity problem that I have mentioned. So there's an accumulation of inhibitory cells to make things worse. So all of these things happen in aging, and they do not help productive immunity. So the essential challenge that we have in trying to understand immune aging is, can we define the mechanisms that are behind these intrinsic and population-based defects of immune aging? Can we figure out which things happen first, the primary changes compared to some compensatory changes that could occur later? And also, can we rationally intervene? That's the most important question down the line, at the most effective point, to make things better, to correct immune senescence and to improve immune defense in the old age. So I will tell you now about our research. What do we do to try to understand what's going on and to try to correct it down the line? One of the bugs that we study is the West Nile virus. This is the virus that in the US really was not known to the population until '99, but then it came and it very rapidly spread through the population. It infected a lot of birds. Normally it's the virus that infects the birds. But the reason why we study is that it has this particular death curve or mortality curve. You can see here that people over 75 were dominantly the ones that were dying if infected with this virus. And this is the typical seasonal outbreak in one of the states of the US. And those seasonal outbreaks have happened since '99 every year since, and this year, 2012, looks like to be a particularly bad year because mosquitoes have been multiplying really well. So I already alluded to this now, that this is the virus that's transmitted by mosquitoes. It naturally really circulates between mosquitoes and the birds. An infected mosquito would bite the bird will get the infection, the virus would replicate like crazy in its blood, and the new uninfected mosquito can come feed on that bird, pick it up, and then transmit it to the next host. This virus belongs to the family of flaviviruses, which has other famous members such as dengue and yellow fever. It's a small enveloped RNA virus. The important point, though, is that this mosquito that has fed on an infected bird can now go and bite any other organism and probably get him sick of the West Nile virus. Certainly mice, horses, other domestic animals, and humans are all very highly infected when bitten by an infected mosquito. But they do not transmit the virus any further, and the mosquito cannot refeed on any of these hosts because they never replicate the virus to high levels in the blood. They can still die of the virus, and that is the problem. And one of the main model systems that we use, and we use other model systems such as monkeys and humans, but here we're going to use mostly mice. This is the C57 Black 6 mouse. We use adults that are about 4 to 6 months of age, and our old mice are senior citizens. They're typically over 18 months of age. Normally, these are the mice that live up to 30 months of age, but they start really dying off around between 20 and 24 months. And the reason why we study this infection in mice is that when you infect old and adult mice with the same dose of the virus, you can see that some of the adults die, but most of them survive. Unlike that, most of the old mice will not survive, they will die, and very few of them will survive. So that is now our own research. We also use a bacterium. So this is the bug that's called Listeria monocytogenes. This is the bug that will cause alimentary infection. You can get it through food. And again, adult mice survive much better than the old mice, which die pretty quickly. So we're studying the immune response to these two infections that show pronounced age-related sensitivity. And so what I will show you now is that as you initiate the immune response to either of these two infections, the dendritic cells don't pick up the antigen as well when you're old, and they don't mature as well because they haven't picked up the bug very well. And so there is a problem right here in the microbial capture. There's a problem here in the maturation of the dendritic cells. But also when they move to the lymph node and start talking to the T cells themselves are not present in sufficient amounts. There's not enough of the naive cells here. The precursors are affected. Also, the way that these cells now get the signal and mature and are supposed to become the defenders of the organism is impaired. So their effector immunity against the virus is not nearly as good as it used to be. So in the process of studying these infections, we very often use the experiments like these where we would transfer some of the cells into another mouse. So we take one mouse, we isolate some cells, and we put them into another mouse. And when we started these studies, we wanted to ask a fairly simple question. Is there something wrong with the environment in the old mice, or is it only the T cells that are a problem in the old mice? And we came up with the answer that both of them are a problem. Let me tell you about how we found out about the environment. So we've taken some indicator mice that have genetically modified T cells that all respond to one piece of the bug that we can use. And we took young cells from the young mouse here. So we wanted to see how these young cells would function when we put them into a young mouse or into an old mouse. So we're now varying the environment and we're looking at how they would respond. So when we do that experiment and we transfer these cells, we look at how well can they expand when we give these old or adult mice the infection. So the cells that we're looking at are all young, but they're being now immunized and they're encountering their bug either in an old environment or in an adult or young environment. And you can see here that in the adult environment they expand at least threefold better than they do in the old environment. So that's all telling us that there is a problem in the environment. When we do a more precise experiment to see what happens, we see that in the old mice, these cells now are measuring their ability to divide, and this is what's represented here on the y-axis. We're looking at the old mice. They start to divide kind of at the same time as in the young mouse, but they don't nearly reach the same height in the peak, meaning that their expansion is curtailed in the old mouse, in the old environment. So we've done a lot of experiments that I will not detail here, but they told us that initial steps in dendritic cell sensing and activation of T cells all deteriorate with aging. We found that there is a particular subset of dendritic cells that's called CD8 alpha positive dendritic cells. They are underrepresented during this initial priming. Other cell subsets did not show any difference, and that was very interesting because it has been known that these particular dendritic cells are essential to start the immune response, in this case against Listeria. When we look at how many precursors of these dendritic cells are there in the old mice, we find out that they're reduced. We have tricks and ways and means in which we can try to mobilize them, and we can mobilize them in part to look better, but that doesn't really correct the defect that we're looking at here. The dendritic cells also, when we examine them carefully, are showing reduced uptake of the bug, and that makes the T cell priming very wimpy because they haven't been really activated to a sufficient extent to present enough molecules of the bug to the T cell, and they're not also making all of the other juicy factors and molecules that the T cell needs to see in order to be kicked into activation. We've also shown that there are some of the uptake problems that could be extrinsic to the old cells, again, to the old dendritic cells, meaning that there's something in the environment that's preventing all dendritic cells from picking up the antigen as well as it should. Well, so if we move on now and we ask a different question, now if we ask, okay, so if we make the pickup of the antigen and antigen presentation normal, are the T cells also part of the problem? So normally, as I told you, this dendritic cell, shown here as the APC symbol that stands for the antigen presenting cell, is talking to the T cell once it picks up the pieces of the bug. And the piece of the bug here is shown by this very tiny little red piece on the yellow molecule on the APC. That's recognized by the T cell receptor on the T cell. T cell needs to get all of these other signals that are shown here. So there's a multi-channel communication really happening between the T cell and the APC. As a consequence of this contact, the T cell will come up. It will start putting up the receptor for certain growth factors, like the IL-2 receptor. It will multiply greatly. I told you that one cell will give 1 million cells down the line. And then it will also differentiate into the cell that now is armed and dangerous and that can go into the tissues and it can secrete different cytokines such as type gamma interferon or type 2 interferon. It can also secrete the molecules that will directly kill the neighboring cell, so it can have this cytotoxic effect. And the bottom line here is that all of these steps are defective when we grow old. They may not be drastically defective. They could be affected by, they could be 10% decline or 30% decline, 40% decline, but it's a defect against a series of successive steps. And so many subtle or mild differences in the end produce a major loss of protective function. How does this look really in practice? So when we examine our response against the West Nile virus here, we can see here that in panel A that old cells have many fewer T cells that are specific for the virus. This is what we're measuring on the y-axis here. This is the percentage of the tetramer positive cells that will see the virus. And tetramer means just that we're using certain molecules that are tetrameric, that can stain antigen-specific cells. If we count the absolute numbers, which is more relevant than just scoring the percentages, we now see that there is about 3 to 4-fold decrease in the absolute number of these cells. But when we ask now how many of these cells can actually perform an antiviral function, the answer is relatively few when we're old. Almost every single cell can do that in the young organism, but only about half of these cells can really make interferon gamma in the old organism. And so that tells us that even of the cells that are specific for the virus, many of them are really not able to fight it off. When we take this analysis another level deeper, other important things about the T cells is how many functions can a single T cell do? Generally, you're better protected if your T cells can do a number of different functions. If a single cell can make interferon gamma, make TNF alpha, which is another mediator, it's tumor necrosis factor, which also inhibits viral replication. It can make lipid granules, molecules that will destroy the neighboring cells. It can have this killer function that I told you about. And so if we compare how many cells are polyfunctional, can do multiple things in the old and the adult, the answer is that the old do only about half as well as the adult. When we now also measure how much of each of the molecule is expressed per cell, so how well armed is each of the cells, we find out that there are, again, successive declines by anywhere between 20 and 50% in terms of the amount of the molecule per cell that these cells have. So that means that once they get to fight off the virus, they will have only half of the reserve that their adult counterpart had. So that's all telling us that there are multiple defects. And we found the same defects and the same problems for CD4 cells in the West Nile infection, as well as for the CD8 cells in the Listeria infection. Then we wanted to know this problem in numbers. Is this because the cells don't proliferate as well? Is this because they die too much? What's going on? And so we've done this one experiment where we've looked very carefully, in vivo, how these cells proliferate when you give them an infection. And what we found out is, what you see in this particular slide, is that both adult and old T cells really start to proliferate at the same time. They enter into the proliferation at the same time. What they don't do the same way is they don't accumulate to the same extent. So that there's about 2.5-fold more adult cells compared to the old. And if we want to find out how many divisions did each of the cells undergo, and we can do that by using the in vivo label called BRDU, which is what we're using here. Again, we see that adult cells must have proliferated at least 3 times more than the old cells, based on the intensity of the label per cell. Old cells also do die more than the adult cells, and that's the data that I'm not showing you here in the interest of time. What I want to show you, though, is once these defects are all on display, how does that affect your ability to fend off the infection? So I told you that the West Nile virus is this virus that infects old people and makes them die. It does so by getting inside the brain and really destroying the central nervous system. And so what we did here is we've looked in the brain, and this is not a very clear slide, but what I can tell you is just look at the differences. What we find here is that when we just count the total lymphocytes in the brain, there's fewer in the old compared to the adult. This is the difference. There's roughly about 2 to 3-fold less total lymphocytes in the old brains compared to the adult brains. If you now look at how many of them are CD8 or CD4 T cells, there's another decline in the old. Now when we ask how many of these cells are specific for the virus, there's another decline here, which is really running between almost a 2-fold loss in the old. And what you get in the end, when we ask how many of these cells are properly armed and how many of them have sufficiently large amounts of molecules that are going to beat the virus back, you end up with another decline. And so when you add all of these declines, there's a decline here, there's a decline here, and sometimes they're 40%, sometimes they're 15%, sometimes they're 2.5-fold. Together though, old mice end up with 20 to about 12 to 20-fold fewer CD8 T cells specific for the virus and properly armed in the brain. And that's a real problem, because if you don't contain the virus at the point where it invaded the brain, you're probably not going to survive. So finally, we wanted to do another experiment to formally prove that our cells, T cells and CD4 and CD8 T cells are really the key to this age-related vulnerability. So we have taken naive old or adult mice, and we purified their CD8 cells. And so then we've transferred them into these test tube mice that don't have their own T or B cells. So when you do that, you're really reading out how much protection can you get from the transferred cells. And so the results were very clear and very conclusive here. The mice that don't get any transfer basically die, and so that, about more than 80% of them die. Sorry. And if you give these mice that don't have their own T and B cells, adult CD4 cells, they're protected quite nicely. If you give them adult CD8 cells, they're also protected very well. But if they get old CD4 or CD8 cells, they're not protected at all. There's no effect really over no transfer of cells whatsoever. So the conclusions from this part of the talk is that old naive T cells show a decline in proliferation and differentiation in response to both viral and bacterial infections. There ends up being fewer effector T cells made, and those that are made are not armed properly to fight infection. This is particularly pronounced in the organs where infection inflicts the highest damage. So in the case of brain for the West Nile virus, that is where you see the biggest differences. So direct transfer experiments also show that old CD4 and CD8 T cells are not protective against infection, whereas the same subsets from the adult T cells are completely protective, or almost completely protective. So I told you that there's a problem in antigen detection and capture and processing. There's a problem in CD8 and CD4 T cell activation. And this is what's illustrated here in the upper part here, where the signals that come to the old cell from the antigen-presenting cell are simply not strong enough to drive proliferation, and we end up with many fewer cells, and some of the molecules are also heavily underproduced. But the other problem that I have mentioned in the introduction is the problem of T cell balance and maintenance and generation. And so this disruption starts when the thymus, which is the organ that produces the T cells, shrinks, and it shrinks very quickly after birth. This is the gland that lives behind your chest bone, and it produces a lot of very diverse cells in youth. And those diverse cells are naive, and they will seed the naive pool. They will live in the lymph nodes, as I mentioned. And with every stimulation, some of them will become memory cells. But what happens in the old is that this thymus mass is drastically reduced. Very few cells get out. Once they get out, they're also not very good at maintaining themselves. Many of them die. Others convert, actually, and become almost like the memory cells. And so that is an interesting process that we have just discovered recently and that we're really interested in studying. So this is the recap of the homeostasis, how we maintain the T cell balance. T cells are constantly being made. They're also constantly being used to respond to infection. There's a set total number of T cells in the body. So therefore, there has to be a balance between the new production, loss by attrition, age-related changes, and changes related to the T cell response to infection. So I told you when there is an infection, there's an enormous expansion. Let's say that you got infected with the flu virus, flu-specific T cells will jump like crazy, multiply. One cell will be divided. It'll make either between 100,000 and 1 million cells. The virus gets eliminated once all of these cells are made. And now the T cells don't need to be there in such high numbers, and therefore many of them are going to be eliminated. Some are going to survive as memory cells, as I told you. They'll be able to respond to the next strain of flu really quickly and to make you less sick. So the survival of these T cells is then mediated by different cytokines, and we'll talk about that later perhaps. Interleukin 15 and interleukin 7 are very important in maintaining these cells. But also persistent pathogens, as I mentioned, can disrupt this balance, because as they reactivate, they will be hitting your T cells over and over again, so they'll be stimulating a large population of T cells. So the next question that we wanted to know is, we've shown that when the dendritic cells are priming T cells, there are problems on both sides of the equation. What we didn't really know is, do we start with the same number of naive cells when we're older? Is there the same number and the same quality of the cells that are supposed to be stimulated and that are supposed to now expand very dramatically, really equivalent between old and the young? So we've done a lot of studies there, and I'm only going to summarize them here in the interest of time. But we found out that old mice lose 2/3 or more of all of their naive cells when they grow older. But when we look at the cells that are still there and that are surviving, they don't survive all in the same manner, and the survival is very selective. We're trying to understand why and what drives it. But at least some of the surviving cells we have found are in a highly activated state. They're really not looking like your bona fide naive cells. The fact that these cells are highly activated could be potentially beneficial, because cells that are immediately activated are also very good at immediately fighting the infection. The problem is that the balance is being lost here, because the ability of these cells to expand and protect is impaired with age. And so therefore, if you lose the ability to expand and proliferate, even if you're able to mount an initial rapid response, if you have a microbe that's going to proliferate very quickly, you're likely to lose that battle, because there's not enough T cells to take care of an expanding population of the viral cells. So that is definitely a big issue and a big problem, and we're trying to now understand how can we better maintain these naive precursors and what we can do to rejuvenate them. But an additional problem that exists in aging is the one that I have mentioned before, and that is the problem of persisting infections. Here in this figure, we're looking at the relative growth curves and expansion curves of the invading microbe and of the immune system. So let's say that we've got infected with the flu virus. The flu virus will multiply very quickly. It will be exponentially expanding. You'll have more and more viral particles everywhere. That will trigger innate immunity at first, and then innate immunity will trigger adaptive immunity. Once antiviral T cells are being made in particular, but also antiviral antibodies down the line, they will start beating this virus back, and they will essentially abrogate its proliferation, and you will not be able to find the virus anywhere else. And that is called sterile immunity, at which point you've eliminated and cleared the virus completely. The cells that remain will contract. I told you that we don't need as many of them at that point, and many of them will turn into memory. And those memory cells will then defend us if the same virus should strike again. But what happens if you have a persistent virus is the virus goes up, innate immunity goes up, adaptive immunity goes up, it controls the virus in the acute phase, but the virus now hides and it goes to different cells where it can become latent, where it can sleep and be dormant and then reactivate when it feels so. And the problem is that this type of viral persistence doesn't really allow these cells that have expanded very well to contract. So there's a problem in contraction. The contraction is not complete, and the virus then reactivates again, and there's another blip where the cells specific for the virus are being stimulated here. And so with every cycle of viral activation, you will have successive stimulation of these cells. And so that successive stimulation of these cells will lead to the growth of the pool of the cells that are specific for this virus. And because you cannot expand indefinitely, there is a concern that these cells might be usurping space and growth factors and maintenance factors from the naive cells, which we have less as we grow old anyway. And the other problem is that these memory cells are trigger happy. They're the cells that can make cytokines at the moment's notice. And so if you tickle their receptors a little bit, maybe they will also play a role in this systemic increase in inflammation that very often accompanies the old age. So let's talk a little more about these persistent herpes viruses. Why are they so important? Well, because in humans, being positive for certain herpes viruses, particularly for the cytomegalovirus or CMV, that was associated with studies of that have shown that mice that have CMV and humans that have CMV tend to have signs of increased immune aging. In humans, that has also been correlated with reduced residual lifespan. In other words, people that have CMV and are old are likely to survive less than people that are CMV negative and are old. And also, when you look at what this may cause and why these people might be dying, one of the things that has been shown very clearly in the publications is that there's an increased incidence of cardiovascular disease in people that have CMV compared to those that do not, even if they're matched for age. So essentially what happens immunologically is that we start with this in youth with these naive cells. Some of them, as they're stimulated, will become memory. Because naive cells are being used all the time and we don't replenish them in the old age, we have fewer naive cells and many more memory cells. But the cytomegalovirus comes in and stimulates the immune system repeatedly. And that results in the generation of this large pool of CMV-specific cells that now clogs the memory compartment of the T cells. And so it can, at least potentially, as I said, take away growth factors, maintenance factors, and also increase susceptibility to new pathogens by interfering with the function of the naive cells. So the overall recap and conclusions from our work is that we have documented cumulative age-related disturbances in T cell homeostasis and function in the old mice, in monkeys and humans. I didn't really present much of the monkey and human data, but our CMV data is largely done in both monkeys and humans. And these alterations change T cell maintenance and homeostasis. They change repertoire balance. The ability to respond to previously unencountered pathogenic challenges compromised. Other problems arise also when persistent infections are present. And the point here is that the treatments would be very different for different types of disturbances that we have identified. So one would want to use vaccination or repertoire rebalancing or thymic rejuvenation or other treatments at different time points. And so let me illustrate that just a little bit. So I told you that when you vaccinate older adults with flu, that roughly only about 1 in 3 is going to have the benefit. So how can we help that? So one of the things that we're doing in the laboratory, we're exploring the concepts of screening. So we're trying to determine how many of these people have youthful-like immune system when we look at how many naive cells they have and how many memory cells and what conditions these cells are in. And so for the people that have youthful immune systems, there will be no intervention needed. You can vaccinate them against flu, and they probably will be those that are 1 in 3 that are having the benefit from the vaccination. The problem is that 2/3 that don't, we need to determine which ones still have a good immunological reserve, but perhaps weak and wimpy responses. So who has the cells that you can potentially stimulate? And then in that case, we need to apply immunostimulation and also improved and tailored vaccines that are tailored around the defects that we see in the old immune system to bypass it. Finally, there will be also a category of people that have little or no immunological reserve. If you lose most of your naive cells and the new infection comes, nothing will happen in terms of your immune defenses. nobody will be there to respond. It's akin to somebody knocking on the door, and if people are not home, they're not going to open the door. So in that case, we really need immune rejuvenation, and that is imperative in this case. And so trying to figure out how to rejuvenate the thymus, how to get better production of lymphocytes from the bone marrow, and how to orchestrate all of that is really the challenge that's upon us. And again, the one where the SENS Foundation is funding some spectacular and very exciting research, not only in our laboratory, but in many other laboratories around the world. So the challenge is really to distinguish between these three groups, to screen them and be able to practically direct them to treatments that will be optimal for the human population in the older adults that really need it. So thank you very much. My name is Dr. Janko Nikolich-Zugich, and I am really hopeful that we will soon have significant contributions in terms of how to treat older adults and how to improve their immune system. And I would like to thank you for listening. I hope that you have learned something. I would also like to thank the SENS Foundation for sponsoring some of the very vital research that we have been engaged in.